What happens in your brain while you dream? If you are settling into bed, pulling the duvet up, or simply pausing for a late-night scroll, that question is the gentle lamp we'll carry tonight. Imagine for a moment that when your eyes close, the lights dim in the house of you, and somewhere backstage, the brain quietly prepares a show. A private cinema where images, feelings, and strange narratives flicker across a screen that only you can see. That theater is what we call dreaming, and tonight I'm going to walk you slowly through its stagecraft, the rhythms that cue the performance, the players that act it out, and the tricks the mind uses to stitch scenes together while most of the rest of you sleeps. First, a brief compass point so we don't get lost in the scenery. When I say dream, I mean the subjective experience, a sequence of imagery, emotion, and sometimes sensation that comes to us during sleep. Dreams can be vivid, bizarre, banal, or terrifying. They might replay fragments of your day, invent wild scenarios, or deliver a melody as if from nowhere. Most people experience them several times each night, even though we remember only a fraction. They are not ghostly messages, they are brain-produced events, a film made of firing neurons, memory traces, and chemical tides. But they are also stubbornly personal, threaded through with emotion and meaning. How do those two truths sit together? That is the question that has fascinated storytellers, priests, and scientists in equal measure. If you enjoy these slow, sleepy science journeys, a small like or a quick subscribe really helps this little channel stay awake. Centuries ago, dreaming was often read as a sign or a message from beyond. The earliest written records we have from Mesopotamia and Egypt show dreams treated as omens or divine visits, documented carefully by scribes and interpreted by specialists. In classical Greece, Aristotle took a more naturalistic tack. He wondered whether dreams might be remnants of sensory impressions, a kind of echo when the body is still but the senses are not entirely shut down. Fast forward to the late 19th and early 20th centuries, and the conversation shifted again. Sigmund Freud cast dreams as the royal road to the unconscious, a disguised fulfillment of repressed wishes. Carl Jung, less interested in repression than in symbols and archetypes, suggested dreams connect us to a deeper collective psyche. Both theories made dreams centrally meaningful, but they also tended to assert stable, single-layered interpretations, a tempting certainty that modern neuroscience resists. Why resist? Because we can now measure brain activity. Where once dreams were divined, they can increasingly be correlated with electrical rhythms and metabolic signals inside the skull. That does not make them mundane. It simply means we can ask new, testable questions about what the dreaming brain is doing. Are dreams tidy metaphors with one true meaning?
The evidence suggests not. Are they emergent properties of biological processes that also have psychological significance? Quite possibly. As we move from myth to measure, we keep both the wonder and the necessary caution. We can describe how the show is staged without insisting it has one fixed script. To understand what the brain does while you dream, we first need to understand the scaffolding called sleep architecture, the repeating pattern that structures a night's performance. Sleep is not a single uniform state. It's a cycle, roughly 90 minutes long in most adults, that repeats several times overnight. Each cycle travels through lighter to deeper stages and back again, and one of those stages, REM sleep, is the one most closely associated with vivid dreaming. The first stage, and one, is the twilight drift. It's that hazy, half-asleep place where images flit in and out like moths near a porch light. People waking from N1 often report fleeting sensations or the feeling of having fallen. The neural signature of N1 shows a move away from clear wakefulness patterns toward lower frequency activity. If sleep stages were a journey, N1 is the lane change onto the motorway. Then comes N2, the workhorse of sleep. This stage occupies the largest proportion of total sleep time for most adults. On an EEG, the cap that records electrical rhythms, N2 features two curious phenomena: sleep spindles and K-complexes. A sleep spindle is a brief burst of rapid activity. Think of it as a tiny internal symbol crash that says, "Keep the gate shut for a moment." Spindles seem to play a role in protecting sleep and in memory consolidation, as if the brain is pressing "do not disturb" while it files certain things away. The K-complex is a lone, large waveform, a brain's audible exhale, which can be triggered by outside noises, but also appears spontaneously. If spindles are bouncers, K-complexes are the sentries who react to a stimulus and decide whether the party can continue. N3, often called slow wave sleep or deep sleep, is the restorative heart of the night. Here the EEG shows slow, high-amplitude waves, the brain's equivalent of a slow, sweeping tide. Deep sleep is associated with physical restoration, growth hormone release, immune system maintenance, and a lowering of metabolic activity. It is also critical for certain types of memory consolidation, especially transferring recent daily experiences into more stable storage. You are harder to wake from N3. If someone drags you out, you come away groggy. Think of N3 as the night's cleaning crew, moving through the house with slow, purposeful strokes. And then, in most cycles, the show pivots into REM, rapid eye movement sleep. REM is the flamboyant stage. The eyes dart beneath closed lids, brain metabolism increases, and the EEG resembles a subset of wake-like activity. It's during REM that we are most likely to have vivid, story-like dreams.
The name tells you one obvious feature. The rapid eye movements are visible and were, historically, the clue that linked dreaming to a specific sleep state. REM typically grows longer in the latter half of the night. Whereas deep sleep dominates the early cycles, REM takes center stage as the night progresses. In total, REM occupies roughly 20 to 25% of a typical adult night's sleep, though that proportion changes with age and individual differences. These cycles are not just boxes on a timeline. They have different chemical environments and circuitry engaged. Neurotransmitter levels shift, systems that promote wakefulness quieten, while others take over and create the conditions for REM's distinctive physiology. The practical implication is that where in the night you wake affects whether you will remember a dream. Wake from REM and you have a better shot at remembering a narrative. Wake from deep sleep and you might have a smell, a mood, or nothing at all. That variability in recall is one reason dreams seem so fleeting. Now let's meet the cast, the brain regions that show up on stage during dreaming. If the architecture is the theater, these are the actors. Some play loud, some whisper, and some go off stage entirely. The prefrontal cortex is our in-house editor. When you're awake, it helps you reason, weigh consequences, and maintain a running inner monologue. During REM, activity in parts of the prefrontal cortex decreases. The editor goes dim. The result is why dream logic often feels uncorrected, bizarre leaps, improbable combinations, and weak critical awareness. It's as though the critic of the script took a break and the improvisers ran wild. The amygdala is the emotional amplifier. It lights up in REM and in dream periods associated with strong feelings. If dreams feel emotionally intense, fear, anger, longing, the amygdala is a key suspect. That activity helps explain why dreams can be vivid emotionally, even if their narrative coherence is thin. The hippocampus acts as the librarian and travel clerk. It helps lay down and retrieve episodic memories. During sleep, especially in non-REM stages, the hippocampus appears to replay patterns of activity that correspond to learning experiences from waking life. In some animal experiments, like those on rodents navigating mazes, hippocampal place cells fire in sequences during sleep that mirror sequences seen while the animal was awake. This replay is one of the main pieces of evidence used to argue that sleep supports memory consolidation. Importantly, though, hippocampal dynamics during REM and non-REM are different, which helps explain why some dreams are fragmentary snapshots of memory rather than coherent replays. The occipital lobe, the brain's visual processing area, is another prominent actor. During REM, occipital activity can increase, and the neural machinery that constructs visual experience is engaged. Imagine the occipital lobe as the film studio, assembling frames and lighting scenes. When it works without the usual external input from the eyes, the images it produces are internally generated, and that's our dream cinema. There's also a set of deep, older structures in the brainstem that orchestrate some of the fundamental features of REM. One such phenomenon is the PGO wave, short for ponto-geniculo-occipital wave, first observed in animals. PGO waves travel from parts of the pons to the lateral geniculate nucleus and then to the occipital cortex, roughly speaking, and are thought to help trigger the visual imagery that characterizes REM. If the brain were a theater, PGO waves would be the stagehand cues, the little lights that flash to signal, now put up the backdrop, and the visual studio starts rolling. Another hallmark of REM is muscle paralysis, known as REM atonia. The brain actively inhibits motor neurons in the spinal cord, so the body doesn't act out the dream. It's a safety mechanism. While the mind rehearses a chase sequence, the legs stay put. The brainstem sends inhibitory signals. Imagine a firm, polite bouncer gently holding the limbs still. Without REM atonia, people can enact their dreams physically, a condition that can be dangerous. But in most of us, the safety lock functions well. What happens when these players act in concert is a production that feels like a narrative, but often lacks the steady criticism and rule-bound logic of waking thought. The prefrontal editor's relative silence, the amygdala's warmth, the occipital studio's vivid output, the hippocampus's scattered filing of recent events, and the PGO cues together create scenes that are emotionally charged, visually elaborate, and sometimes delightfully nonsensical. The mix explains why dreams can feel meaningful, even when their semantics, their literal meaning, are slippery. To ground these mechanisms, let me tell you a few short stories of how dreams have intersected with creativity and science. Paul McCartney famously described hearing the melody of Yesterday arise in a dream. He woke, recorded it, and returned to sleep with new lyrics forming. Mary Shelley's account of Frankenstein begins with a waking dream, an image of a pale student of unhallowed arts, bringing life to a creature. The scenario itself reportedly glimpsed in a dream. These anecdotes are not proof that dreams are magical incubators, but they illustrate how the brain's nocturnal recombination of fragments can produce novel combinations that the waking mind sometimes recognizes as creative. On the experimental side, rodent studies have given us a window into the machinery of memory consolidation. In the 1990s and since, neuroscientists observed that rats running mazes show particular sequences of neuronal firing. Later, during sleep, those sequences replay, and sometimes in compressed form.
The replay correlates with better performance on subsequent maze trials, suggesting a functional role in memory. It's tempting to extrapolate directly from rats to humans, and while the core idea, that sleep supports memory consolidation, is supported in many human studies too, the details are inevitably more complex in a brain with language, abstract thought, and cultural context. Imaging studies in humans add another layer. Functional MRI and EEG work show that REM is associated with increased activity in the amygdala and occipital regions and decreased activity in certain prefrontal areas. That pattern fits the phenomenology: strong emotion, vivid sensation, reduced executive control. But methods matter. EEG gives us excellent timing resolution and core spatial resolution. fMRI gives us better spatial maps but less temporal precision. Neither is a perfect camera for dreams, which are transient and subjective. When scientists say we see increased activity, they mean we observe patterns that tend to correlate with reported dream features, but there is substantial variability across people and across nights. Small sample sizes, noise, and individual differences mean we should be modest in our claims. A closely related topic is dream recall. Why do we remember some dreams and almost none of the rest? Awakening from REM increases recall probability, as I mentioned, but personality, routine, and attention matter too. People who keep dream diaries tend to recall more dreams. It's plausible that training attention to the nocturnal narrative improves memory encoding or retrieval. The so-called continuity hypothesis proposes that our dreams reflect waking concerns and experiences. In other words, day residues often appear in dreams. Studies have found that people often dream about recent events, worries, or preoccupations, which supports continuity, though dreams also recombine and distort these elements. We've covered architecture and the principal players. Now we begin to glimmer at the purposes that researchers have proposed. There is no single agreed answer. Instead, a suite of theories, each with evidential support, attempts to explain different facets of dreaming. Some focus on memory and learning. Some emphasize emotion regulation. Some treat dreams as evolutionary rehearsal of threats. Others propose dreams are epiphenomena, byproducts of other neural processes, without direct function. The debate is lively because dreams are multifaceted. They are both a laboratory for memory processing and a theater for emotion, and their features vary with stage, age, and context. One prominent idea is that dreaming, particularly in REM, aids emotional processing. Evidence suggests that REM may help decouple the emotional charge from the content of memories, allowing one to remember an event with less emotional reactivity afterwards. If accurate, this would position REM as a kind of overnight therapist, not in a conscious, deliberate way, but as a passive recalibration of affect. This theory aligns with observations that after stressful experiences, changes occur in REM architecture and in how emotional memories are consolidated. But causality is tricky. We don't yet entirely know whether REM causes emotional recalibration or merely accompanies processes that do. Another influential perspective treats dreams as part of memory consolidation. During sleep, especially in non-REM stages, but with interactions into REM, Neural circuits appear to replay waking patterns, stabilizing and integrating memories into long-term networks. Dreaming might be a phenomenological byproduct of that replay. As networks are reactivated, fragments bubble into consciousness, stitched together by the mind's associative machinery. This view comfortably accounts for the appearance of recent experiences within dreams and the laboratory demonstrations of replay in animals. It also explains why certain types of learning, procedural tasks for example, benefit from specific sleep stages. Then there's the threat simulation hypothesis, which posits that dreaming evolved to simulate hazardous encounters, allowing the brain to practice threat perception and response in a safe environment. Dreams filled with chases or conflicts could, in this view, be rehearsals that tune our threat detection circuitry. Some cross-cultural analyses suggest dreams often contain socially relevant or threatening content, but the generalizability is not uniform. Many dreams are neutral or even pleasant. The theory remains intriguing, especially from an evolutionary lens, but it probably does not capture the whole story. Activation synthesis theory, a classic neurophysiological account, proposes something more mechanistic. According to this idea, spontaneous brainstem activity during REM, those PGO waves and associated bursts, activate various cortical regions. The forebrain then attempts to make sense of this random activation, synthesizing a narrative from the pieces. The content is therefore largely a byproduct of the cortex trying to interpret internally generated signals. This accounts elegantly for much of the bizarreness seen in dreams, and for why dreams often mix memories with odd imagery. Yet critics argue that activation synthesis treats dreams as mostly meaningless noise, whereas dreams can be highly structured, and sometimes evidently useful for problem solving. Which of these explanations is correct? The honest answer is probably some combination of them. The brain is a multi-purpose organ. Processes that evolved for one function can be co-opted for others. Memory consolidation, emotional processing, and creative recombination are not mutually exclusive.
Different stages of sleep may contribute distinctively to these processes. REM might favor emotional and associative recombination, while non-REM slow-wave sleep favors stabilization and transfer of new memories into longer-term systems. The multiplicity of functions helps explain why interfering with sleep in different ways produces varying cognitive and emotional consequences. If you're still with me, you may be wondering, how do we actually test these ideas? Science uses several complementary approaches. Behavioral studies measure performance changes after sleep or sleep deprivation. Animal experiments allow invasive recordings and manipulations to test causal mechanisms. Human neuroimaging correlates reported dream content with brain activity. Some clever experiments have attempted to bias dream content, for example by presenting sounds or scents during sleep to see whether related images appear in subsequent dreams, and they provide tentative evidence that external cues can nudge dream content without fully overriding the internal dramaturgy. There are also studies of people who experience unusually frequent or vivid lucid dreaming, which offers another window into the neural correlates of self-awareness during sleep. One set of experiments that has captured public attention involves attempts to decode dream content using machines. By correlating patterns of brain activity measured with MRI or EEG with visual categories or features, researchers have begun to predict at a coarse level whether someone is seeing faces or houses, or whether a simple visual category is present. A notable effort by a Japanese research group used machine learning to predict categories of dreamed images above chance in some participants. That work is exciting, but it's crucial to be cautious. The reconstructions are approximate, the fidelity is low, and these methods struggle with narrative, emotion, and idiosyncratic symbolism. We are a long way from reading full dream reports from brain signals. The ethical implications are profound, though, and we would do well to discuss them as these technologies improve. There is also the phenomenon of lucid dreaming, a special case where the dreamer becomes aware they are dreaming and can sometimes exert control over the dream content. Neuroscientifically, lucid dreams appear to involve reactivation of prefrontal regions that are usually less engaged during REM, allowing a degree of meta-awareness. Researchers have even trained lucid dreamers to signal to experimenters by moving their eyes in prearranged patterns while in REM, demonstrating that a form of two-way communication with the dreaming mind is possible. Lucidity opens intriguing avenues for therapy and creativity, but induction techniques are probabilistic and may disturb sleep. We'll return to lucid dreaming and its practical and ethical considerations later.
So far, we've wandered through the theater, met the cast, and sketched several reasons why the show might be running. We have touched on experiments, anecdotes, and brain imaging, but many questions remain open. For instance, how much of dreaming is necessary or merely epiphenomenal? How do different kinds of dreams, the mundane, the bizarre, the recurring nightmare, map onto specific neural patterns? What individual differences determine who remembers their dreams and who doesn't? And how far can technology go in nudging or decoding dreams without violating the most intimate aspects of mental privacy? To begin to answer these, we need to look deeper at particular mechanisms and evidence, to follow up on the actors we've named and watch how they respond in experiments, in clinical cases, and in comparative studies across species. We will look more closely at the neuroscience of lucid dreaming, at parasomnias such as nightmares and sleep paralysis, and at the tantalizing work that tries to read or shape dreams with external cues and machine learning. We will also consider dreaming beyond humans. What does it mean if an animal shows replay during sleep? Does a bird singing in its sleep dream in anything like our sense? And finally, we will circle back to ethics. What does the sanctity of the inner world demand from researchers, clinicians, and technologists who might one day tap that world? Before we move on, let me return to two concrete experiments that illuminate the interplay between memory and dreaming. One line of human research asks whether learning a task changes subsequent sleep architecture. Students asked to memorize lists or motor sequences often show alterations in spindle density or in REM proportions after intense study. Some studies report enhanced performance the next day after sleep, compared with a similar period of wakefulness. A second line of evidence, from rodents, shows replay of neuronal sequences after navigation. Both lines are convergent. They support the idea that sleep participates in memory consolidation, but neither fully explains dreaming's rich phenomenology. Memory consolidation explains why fragments of the day appear in dreams. It does not by itself explain why dreams sometimes engulf us in emotion or invent elaborate, bizarre scenarios. And then there is creativity. Historical anecdotes and some lab studies suggest dreams can foster creative insight. Whether this is because the disconnected associations in dreams allow unusual combinations to surface, or because the different neurochemical environment of REM facilitates associative leaps, is an active subject of inquiry. Creativity in dreams is neither guaranteed nor universal. Like many aspects of dreaming, it is probabilistic and individual. We've covered architecture, key brain regions, and beginning sketches of function.
We've seen how prefrontal quieting, hippocampal replay, amygdala activation, occipital imagery, and PGO cues combine to produce dream phenomenology. We've glanced at evidence that sleep supports memory and emotional processing, and at early efforts to nudge dream content or decode it with machines. There is, however, more to unpack, particularly around lucid dreaming, clinically relevant phenomena like nightmares and sleep paralysis, comparative data from animals, and the murky frontier of dream technology and ethics. Those topics will let us connect mechanism to meaning and science to everyday experience. For now, as you perhaps float on the edge of sleep or sit quietly in a chair, consider this. Your brain stages an astonishing nightly program with its own rhythms, its own cast of characters, and its own backstage bureaucracy. It does this in service of functions we are still mapping, and in the process, it produces experiences that can surprise us, distress us, console us, or sometimes light the first spark of an idea. What we can reasonably say is that dreaming is neither pure mysticism nor mere epiphenomenon. It is a biological process with psychological resonance, messy and beautiful in equal measure. Next, we'll pick apart some of those theoretical proposals in more detail. We'll look at the laboratory evidence for memory consolidation and emotional regulation, and then explore lucid dreaming, nightmares, and parasomnias, and the comparative data from animals that hint at dreaming's evolutionary roots. We'll also take a careful look at dream decoding technologies and the ethical questions they raise, because the more we learn about the sleeping brain, the more we owe it to its privacy. But before that, let me ask you something to keep in mind as you listen. When was the last time you were surprised by a dream? Did it feel more like a memory, a film, or an absurd comedy? Reflect on that and hold it lightly. The patterns we'll discuss next will often find mirrors in your own nightly cinema, sometimes a clear reflection, sometimes an odd, distorted echo. We left off with a question to hold lightly. When were you last surprised by a dream? Keep that image in your mind as we take a closer look at the evidence that tries to explain not just how dreams are made, but why they might be useful, and what we still do not know. One major strand of research treats sleep as the nightly workshop where memories are molded. Here, the evidence is convergent but nuanced. Behavioral experiments have repeatedly shown that sleep improves certain kinds of learning. After a period of sleep, people often perform better on tasks involving motor skills or on tests that require integrating new information. The sleep-dependent boost is not uniform. Some forms of memory profit more from slow-wave sleep, others from REM.
but the pattern points to sleep playing an active role in stabilizing and reorganizing what we learned during the day.
The neuroscientific story beneath those behavioral findings is where the hippocampus replays its cards. In animal work, the replay phenomenon is perhaps the clearest demonstration. Neural sequences that map to a rodent's path through a maze are reactivated during subsequent sleep. These replays often occur in compressed time and can be coordinated with slow oscillations and spindles that sweep through the cortex. The interpretation many researchers favor is that the hippocampus rehearses recent events and communicates them to cortical networks, a transfer that gradually embeds fragile new memories into more stable cortical circuits. Dream fragments, in this view, might be the subjective shadow of that replay. Sometimes we experience reactivated elements as images or scenes. But memory consolidation alone does not capture emotional tone. That is where theories of emotional processing come in. Several studies suggest that REM has a particular relationship with affect. After emotionally charged experiences, REM density and architecture can shift. Some experiments show that REM-rich sleep attenuates the affective charge of a memory, allowing a calmer recollection the next day. Think of it as the brain gradually disinfecting a sting. The memory content remains, but the immediate pain diminishes. It is important, though, to be cautious about causality. Does REM directly dampen emotion, or does it support a broader set of processes, of which emotional recalibration is one? Evidence suggests the latter. REM is probably a favorable milieu for reprocessing affect-laden memories, but it likely interacts with daytime cognitive appraisal and social support in shaping emotional outcomes. Another idea, the threat simulation hypothesis, takes an evolutionary turn. From this perspective, dreaming is a rehearsal system for dealing with danger. Dreams filled with chases, confrontations, and escape behaviors could have once provided a low-cost environment to exercise threat detection and response. Cross-cultural studies indicate that socially relevant threats are common dream themes, but the distribution of threatening content across all dreams is not overwhelmingly dominant. In short, threat rehearsal may be one of several functions that dreaming supports, particularly in species with complex social lives, but it is unlikely to be the whole story. And then there is activation synthesis, a mechanistic, parsimonious account that leans on neurophysiology. Spontaneous activations from the brainstem during REM travel upwards, exciting cortical areas without external sensory input. The cortex then attempts to weave those activations into a coherent narrative, the synthesis. This framework explains the bizarreness of many dreams and why dreams often repurpose memories into odd juxtapositions. Critics argue that this hypothesis risks reducing dreams to noise, but it remains powerful because it directly links known physiological events to phenomenology.
The most defensible position, perhaps, is a pluralistic one. Dreams may meet multiple adaptive and byproduct criteria simultaneously. Sleep consolidates memories, REM provides an emotional workshop, activation events yield raw material that the cortex reorganizes, and occasional threat-like scenarios provide practice. The sleeping brain is not a one-function tool. It is a multi-purpose studio that can perform several tasks at once. How do researchers test such claims? It helps to look at the ingenuity of experimental design. One family of experiments asks whether externally supplied cues can bias dream content. Play a sound associated with a learning task while somebody sleeps, or waft a scent that accompanied a daytime experience, and later you may find that related elements are more likely to appear in dreams. These experiments are careful and modest. Cues can nudge, not dictate dream content. They suggest the sleeping brain remains responsive to some forms of external input, even when it is otherwise operating on internally generated activity. Other experiments aim to establish two-way communication with the dreaming mind. In specialist labs, lucid dreamers, people who have either trained or spontaneously learned to become aware inside their dreams, have been instructed to perform prearranged eye movements to signal that they are lucid. Because the eyes move under the closed lids while the rest of the body is paralyzed during REM, these eye movement signals can be detected in polysomnography and matched to reported dream content upon awakening. Those studies are proof of principle. They show that a dreaming subject can, at least briefly, exchange information with the waking experimenters. The technique opens fascinating possibilities for studying dream content more directly than simple post-awakening reports allow. And then there is the realm of neuroimaging and machine learning, where researchers try to decode the neural signatures of visual experience. Using fMRI or high-density EEG, teams have trained algorithms to associate patterns of brain activity with categories of images, faces, houses, common objects, while subjects are awake and viewing stimuli. Some groups have extended this idea to dreaming. They build models from waking perception and then apply them to sleep data, testing whether predicted categories align with dream reports. The results are intriguing. Classifiers can sometimes predict broad categories above chance and reconstruct rough visual features. One research group reported category predictions above chance in sleepers, which drew considerable attention. Yet the reconstructions are low fidelity. They capture broad strokes rather than detailed narratives, and they often rely heavily on statistical priors and the limits of the recording method.
It is essential to emphasize the limitations.
EEG has superb temporal but blunt spatial resolution. fMRI maps space rather well, but blurs time and requires an immobile subject inside a noisy magnet. Dreams are fleeting, highly individual, and rich in personal associations that are not readily mapped to standardized visual categories. Machine learning models can pick up statistical regularities, but they cannot read private meanings or emotional subtleties. In other words, dream decoding is an exciting technological frontier, but not a crystal ball. The ethical questions that follow, however, are already urgent. Before we move fully into ethics, let us linger on lucid dreaming a little longer because it offers a striking window into altered states of consciousness. Lucidity is, quite simply, the recognition within a dream that you are dreaming. For some people, this recognition is accompanied by partial restoration of prefrontal activity. That is, regions involved in metacognition and self-monitoring flicker back on, and with that can come intentional control over parts of the dream. Neuroimaging studies suggest that lucid REM shows a hybrid pattern, many features of REM coupled with increased frontal activation relative to ordinary REM. That hybrid state makes lucid dreaming particularly interesting to scientists interested in the neural correlates of awareness. Practically, people attempt to foster lucidity using several widely discussed techniques. The first is reality testing. During the day, you periodically ask, "Am I awake?" and perform a simple test. Try pushing a finger through your palm, or look at a text twice to see if it changes. The idea is to build a habit that, with luck, carries into dreams and triggers the insight that you are dreaming. Another method, called mild, mnemonic induction of lucid dreams, involves waking yourself after several hours of sleep, rehearsing the intention to recognize a dream, and then returning to sleep with that intention active. Wild, wake-initiated lucid dream is more ambitious. People aim to keep awareness during the transition from waking to sleep, stepping consciously into a dream. All these methods increase the probability of lucidity in some people, not guarantee it. They can also fragment sleep if used excessively. Waking up repeatedly or ruminating too much at night may reduce sleep quality. So the promise of lucidity comes with a practical caveat. Techniques are probabilistic and may disrupt restful sleep if practiced without balance. From a research standpoint, trained lucid dreamers have enabled those eye movement signaling studies I mentioned, and they have allowed researchers to probe how self-awareness and volitional control map onto neural activity in sleep. There are potential applications in therapy, notably for recurrent nightmares, and in creativity.
Some practitioners deliberately use lucid dreams to practice skills, confront phobias within a safe simulated environment, or explore imaginative scenarios. Yet these uses remain an active area of study, and any therapeutic application ought to be undertaken within a clinical framework rather than as a do-it-yourself cure. That brings us naturally to nightmares and parasomnias, which are demonstrations of how dreaming can sometimes harm rather than help. Nightmares, typically occurring in REM, are dreams that evoke strong negative emotions and can awaken the sleeper. Night terrors, by contrast, are usually non-REM phenomena, sudden episodes of arousal with intense autonomic responses, often with little or no recall of specific dream content. Sleep paralysis occurs when REM atonia persists while consciousness returns. The mind wakes before the body, leaving a person temporarily unable to move, often accompanied by vivid hallucinations and a profound sense of fear. These experiences can be terrifying and in some cases contribute to disrupted sleep and daytime impairment. There is a clear link between trauma and disturbed dreaming. People with post-traumatic stress disorder frequently report recurrent, distressing dreams that replay aspects of the traumatic event. Research suggests that alterations in REM and in the way memories are processed during sleep may contribute to the maintenance of such symptoms. One empirically supported approach for distressing dreams is imagery rehearsal therapy. The patient, usually with a clinician, rehearse altered or re-scripted versions of their nightmares during waking hours to reduce frequency or intensity. The results are promising for some sufferers, yet it is crucial to state that such methods should be administered by trained professionals. It would be inappropriate and potentially unhelpful to suggest self-guided fixes for complex clinical presentations. Pharmacology can also interact with dreaming. Certain medications suppress REM and thereby reduce dream vividness or frequency. Others may provoke vivid dreams as a side effect. Again, these are clinical matters. Any decision about medication must occur with a healthcare professional, and sleep architecture alterations often have trade-offs. Suppressing REM might reduce nightmares in the short term, but have other cognitive or mood consequences over the long term. Let us now look beyond humans. Do animals dream?
The short answer is evidence suggests they do, though what they dream about and what dreaming means for different species are open questions. The classic rodent maze replay experiments hint at memory-related reactivation during sleep in rats. Birds, notably songbirds, show rehearsal of song patterns during sleep. Playback studies indicate that neural activity during sleep contains the signatures of song learning.
Mammals with complex social behaviors, dogs, primates, exhibit REM and may show behavioral signs suggestive of dreaming, twitching, vocalizations, limb movements that are sub-threshold to full enactment because REM atonia is mostly intact. Interpreting animal sleep as dreaming requires caution. We cannot access subjective report in animals. We infer phenomenology from physiology and behavior, but the continuity of certain neural mechanisms, sleep stages, hippocampal replay, REM-like EEG signatures across many taxa suggests evolutionary conservation of processes that support memory, learning, and perhaps emotion. Whether a sleeping bird rehearses a melody in anything like our conscious sense of playback is unknowable. Still, comparative work enriches our thinking about why sleep and dreaming evolved, and emphasizes that dreaming is not a peculiarly human oddity, but part of a broader biological repertoire. This biological repertoire is now under techno-scientific scrutiny. What happens when machines meet dreams? The last decade has seen steady if cautious progress in so-called dream decoding. The general approach pairs brain recordings with machine learning models trained to associate patterns of activity with perceptual categories during wakefulness, and then applies those models to sleep data. Some studies have produced crude reconstructions of visual content, or predicted broad categories of dreamed images with better-than-chance accuracy in some participants. Likewise, sensory cueing during sleep, a form of targeted memory reactivation, can bias dream content, and in some cases enhance subsequent memory for the cued material. It is tempting to imagine a near future of high-fidelity dream reading, a device that plays back your night's narrative like a video. That future is not imminent. Current techniques are limited by multiple factors: the granularity of the recording method, individual variability in brain anatomy and dream imagery, the probabilistic nature of machine learning predictions, and the subjective, semantic richness of dreams that resists simple visual category labeling. Where progress is tangible is in modest, domain-specific reconstructions, and in devices that can subtly influence dream content, for example, nudging a repressed memory or calming a recurring nightmare through cueing. Even these uses demand careful ethical guardrails. Which brings us to neuro rights and privacy. If a brain signal can be correlated with certain dream features, who owns that data? How do we safeguard consent for an experience that is private and often emotionally charged? Could employers, insurers, or third parties misuse dream data? These are not speculative concerns, they are immediate ethical questions as companies develop wearable sleep trackers that claim to analyze sleep stages, and as researchers explore more ambitious decoding algorithms. The debate must balance potential benefits, therapeutic interventions for nightmares, diagnostic insights into psychiatric conditions, creative tools for artists, against risks of intrusion, misinterpretation, and coercion. A few guiding principles seem sensible. First, consent must be informed and ongoing. Participants should understand what data are collected, how they will be used, and what inferences can and cannot be made. Second, technical limitations should be transparently communicated. Overclaiming accuracy risks both ethical and scientific harms. Third, there should be legal and institutional protections against the unauthorized use of neural data. The inner life deserves legal recognition. Some jurisdictions are already debating neuro rights frameworks that would extend privacy protections to brain data. Such discussions are a necessary part of responsible science. There are also practical design choices that can reduce risk, favoring opt-in designs, keeping raw neural data under the control of the user, and prioritizing applications that demonstrably improve health outcomes. For instance, a wearable that aids a clinician in treating recurrent nightmares under strict oversight is ethically different from a commercial app that offers dream interpretation and sells users nocturnal data to advertisers. Looking ahead, what realistic, ethically defensible possibilities might we expect? One avenue is targeted dream incubation for therapeutic ends, pairing benign cues with therapeutic imagery to reduce nightmare frequency under clinician guidance. Another is wearable technology that helps people improve sleep hygiene by accurately tracking sleep stages and offering personalized recommendations to aid restorative sleep. From a creative angle, carefully controlled lucid dreaming protocols might help artists and writers explore novel combinations of imagery. All these futures require rigorous validation, replication across diverse populations, and societal conversations about acceptable uses. There are also scientific frontiers, better models that integrate temporal dynamics and individual variability, hybrid sensors that combine EEG with non-invasive measures of autonomic state, and collaborative platforms where dream reports, ideally anonymized and consensually shared, can be matched with physiological data to improve decoding models. The cautionary note is that any technical advance must be matched by ethical foresight. Technology is a tool. How we wield it determines whether it becomes a balm or a breach. As we draw this tour to a close, let me return to the core question behind the title we began with: what happens in your brain while you dream?
The short narrative is this: sleep stages orchestrate different physiological milieus. Specific brain regions shift their activity in distinctive ways. Memories are replayed and restructured. Emotions are revisited and often recontextualized. Sensory cortices produce internally generated imagery, and brainstem cues help time and shape the spectacle. Layered on top of these mechanisms are functions and byproducts that range from memory consolidation to emotional processing, from creative recombination to possible evolutionary rehearsal. The result is an experience that feels both intimate and puzzling, intensely personal cinema produced by a biological production studio. Yet it is equally important to close with uncertainty. Many of the mechanisms we discussed remain incompletely mapped. How subjective meaning arises from patterns of firing and chemical gradients is still deeply mysterious. Dream decoding, while promising in narrow strokes, cannot yet reach the narrative, emotional, and symbolic richness of a lived dream. Techniques to induce lucidity or to nudge dreams are probabilistic and must be used thoughtfully. And as technology and science advance, ethical frameworks must advance with them so that the inner landscape remains a space of sanctuary, not exploitation. So tonight, as you settle or scroll, as you tuck in or linger, remember that your brain is staging an activity both ancient and urgent. It is rehearsing, repairing, recombining, and sometimes confronting. It is also keeping a private ledger of associations that only you can fully read. That privacy matters. It deserves both our scientific curiosity and our protective care. If you've enjoyed this slow, sleepy exploration, I'd love to hear where you're watching from and whether this kept you awake or helped you nod off. Tell me in the comments. From the theater of your head to the streets outside, thank you for sharing a little of your night with me. Sleep well. May your dreams be strange in the best possible way.